Hello, welcome to the Creative Proteomics presentation on lipidomics. Today we will delve into the fascinating field of lipidomics, exploring what lipids are, the different types of lipids, the methods used for lipidomics analysis, and the applications of lipidomics in various areas of research and industry. So let's dive right in. Lipids are diverse organic compounds crucial to various biological processes. These hydrophobic molecules are composed of carbon, hydrogen, and oxygen, making them insoluble in water but soluble in organic solvents. Lipids are essential for cell membranes, providing structure, stability, and regulating substance flow to maintain cellular integrity. They also serve as a concentrated energy source stored in adipose tissue to be used during fasting or high-energy demands. In cellular communication, lipids act as signaling molecules, regulating cell growth, inflammation, and immune responses. They aid in the absorption and transport of fat-soluble vitamins A, D, E, and K, ensuring their availability for physiological processes. Lipids also provide insulation and protection. Adipose tissue helps regulate body temperature and protects vital organs, while lipids form a protective layer around nerves for efficient nerve impulse transmission. Lastly, lipids are precursors for hormone synthesis, including steroid hormones that regulate reproduction, metabolism, and immune responses. Lipids can be classified into several major categories based on their chemical structure and biological function. Fatty acyls are the simplest form of lipids, composed of a hydrocarbon chain with a carboxylic acid group at one end. They serve as building blocks for more complex lipid structures. Glycerolipids consist of a glycerol molecule linked to fatty acids. They are abundant in biological systems and act as energy storage molecules. Glycerophospholipids, another important category, have a glycerol backbone, two fatty acid chains, a phosphate group, and a polar head group. They are major components of cell membranes, contributing to their structure and functionality. Sphingolipids feature a sphingosine backbone and play crucial roles in cell signaling and membrane structure. Examples of sphingolipids include ceramides, sphingomyelins, and glycosphingolipids. Sterols, such as cholesterol, have a distinct four-ring structure. They are integral to cell membranes and serve as precursors for the synthesis of steroid hormones. Prenols are derived from isoprene units and play various roles in protein modification, electron transport, and hormone synthesis. Saccharolipids combine lipid and carbohydrate components. They are found in certain bacteria and influence cell-cell interactions and membrane stability. Polyketides are complex lipids synthesized by organisms. They exhibit diverse structures and have various biological activities, including antibiotic and anti-cancer properties. Understanding the diverse array of lipids present in biological systems is crucial for unraveling their functional roles. Lipids play essential roles as signaling molecules, energy sources, and components of cell membranes. Their dysregulation has been implicated in numerous diseases, including metabolic disorders, neurodegenerative conditions, and cardiovascular diseases. To gain a comprehensive understanding of lipid function, it is imperative to employ sophisticated lipid analysis techniques. Now that we understand the basics of lipids, let's explore the methods used for lipidomics analysis. I would like to discuss two important techniques used in lipidomics analysis: gas chromatography-mass spectrometry and liquid chromatography-mass spectrometry. GCMS is a powerful analytical method that allows us to separate and analyze volatile and semi-volatile compounds, including certain lipid classes such as fatty acids. It utilizes gas chromatography to separate the compounds based on their vapor pressure and boiling points, followed by mass spectrometry for their detection and identification. GCMS is particularly useful for fatty acid profiling, quantifying fatty acid compositions, and analyzing volatile lipid metabolites. It finds applications in various fields such as lipid metabolism studies, food science, and clinical research. LCMS offers a broader scope and versatility compared to GCMS. It enables the separation and identification of a wide range of lipid classes based on their physicochemical properties, such as hydrophobicity or charge. Liquid chromatography is employed to separate the lipids, followed by mass spectrometry for their detection and structural characterization. LC-MS provides high sensitivity and specificity in analyzing lipids, allowing for a comprehensive lipidomics analysis. It can provide detailed information on lipid molecular species, including their fatty acid composition, positions of double bonds, and modifications. LC-MS is widely applied in lipidomics research involving lipid metabolism studies, lipid biomarker discovery, lipid-related diseases, and metabolomic studies. To perform a lipidomics analysis, researchers typically follow a general workflow consisting of several key steps. The first step in lipidomics analysis is sample preparation. It is crucial to ensure the proper handling and processing of samples to obtain accurate lipid profiles. This includes carefully selecting and preparing the appropriate sample type for lipid extraction. Lipid extraction plays a vital role in lipidomics analysis. Several methods are commonly used, including solid-phase extraction, purification, using thin-layer chromatography and liquid chromatography, acid-alkaline hydrolysis, and derivatization with additives or signal enhancers. After lipid extraction, the next step is instrumental analysis. Several techniques are employed in lipidomics analysis, including shotgun analysis, liquid chromatography-mass spectrometry, and gas chromatography-mass spectrometry. Different mass spectrometers such as linear ion trap, time of flight, triple quadrupole, quadrupole time of flight, and orbitrap are used in lipidomics analysis. Additionally, various ion sources are utilized in lipidomics analysis to facilitate ionization and ion transmission to the mass spectrometer. Commonly used ion sources include electrospray ionization, matrix-assisted laser desorption ionization, and atmospheric pressure chemical ionization. Finally, data processing and analysis are essential steps in lipidomics analysis. This involves interpreting and extracting relevant information from the vast amount of data generated during instrumental analysis. Advanced software tools and bioinformatics approaches are employed to identify and quantify lipid species, perform statistical analysis, and interpret biological significance. There are two main strategies: untargeted and targeted lipidomics. The goal of untargeted lipidomics is to comprehensively analyze the lipid composition within a sample without predefining specific lipid targets. This approach allows for a broad and unbiased exploration of lipid species present in the sample. Untargeted lipidomics analysis is particularly useful in discovering novel lipid biomarkers, identifying lipid profiles associated with diseases or treatments, and elucidating lipid metabolism pathways. It provides a holistic view of lipidomic changes in a system and can uncover unexpected relationships and correlations. Unlike GC-MS, which is primarily limited to volatile compounds, LC-MS can analyze a much broader range of lipids, including polar lipids, phospholipids, and sphingolipids, which are not amenable to GC analysis. One of the strengths of untargeted lipidomics lies in its high-resolution parent ion selection. Leveraging techniques such as orbital trap mass-to-charge ratio resolution combined with liquid phase time resolution, we can precisely select and analyze lipid species. This capability enables us to detect and identify a vast number of lipid species with the potential for up to 9 million different events in a single sample. This comprehensive profiling allows us to capture a detailed snapshot of the lipidome. However, it is important to acknowledge the limitations of untargeted lipidomics analysis. Firstly, this approach is most effective in detecting striking changes in lipid profiles. It may not be as sensitive in identifying subtle alterations where the change is less than 30% in most cases. Therefore, untargeted lipidomics is particularly useful when investigating significant and pronounced shifts in lipid composition. Furthermore, it is crucial to validate the profiling results obtained through untargeted lipidomics. While this technique provides valuable insights into the lipidome, further experiments and targeted analyses are required to confirm the identified lipid species and their biological significance. Validation ensures the reliability and robustness of our findings. When lipid molecular conformation is performed, targeted lipidomics analysis is required. Targeted lipidomics aims to quantitatively measure and analyze a specific set of lipid species or classes. Unlike untargeted lipidomics, which captures a wide range of lipid species in an unbiased manner, targeted lipidomics allows for a more precise and focused investigation of specific lipid molecules. This approach is particularly valuable when studying lipid alterations associated with certain diseases, metabolic pathways, or cellular processes. The scope of targeted lipidomics is determined by the specific lipid classes or species of interest. Researchers can tailor their analyses to examine specific lipid classes such as phospholipids, glycerolipids, or sphingolipids. Furthermore, within each class, targeted lipidomics can provide insights into individual lipid species or their specific modifications, such as fatty acyl chain length or degree of saturation. To achieve targeted lipidomics, researchers employ various analytical techniques, including mass spectrometry-based approaches. These techniques offer high sensitivity, specificity, and the ability to quantify targeted lipid species accurately. Notable methods include multiple reaction monitoring and parallel reaction monitoring, which enable precise quantification of specific lipid species of interest. By combining targeted lipidomics with advanced instrumentation, such as triple-quadrupole mass spectrometry or hybrid quadrupole Orbitrap mass spectrometry, researchers can achieve high-resolution and accurate quantification of targeted lipid species. These cutting-edge technologies provide robust and reliable data, allowing for precise measurement of lipid changes in different biological samples. The selection of specific lipid species or classes is facilitated by the remarkable capabilities of high-resolution parent ion selection. By multiplying the liquid phase time resolution, quadrupole MZ resolution, and Orbitrap MZ resolution, we can estimate the potential selection capacity of targeted lipidomics. For instance, let's consider values of 150 for liquid phase time resolution, 1000 for quadrupole MZ resolution, and 2000 for Orbitrap MZ resolution. Multiplying these values yields an impressive 3 billion possible events within a single sample. This immense selection capacity allows researchers to precisely target and analyze a vast number of lipid species of interest. Upon completion of lipidomics experiments, the next step is to process and analyze the acquired data. This involves a series of computational and statistical approaches to transform raw data into meaningful results. Let's explore the key steps involved in lipidomics data analysis. First, let's start with the raw data. In lipidomics, we work with data obtained from analytical instruments, including mass spectra, retention times, and metadata. This raw data serves as the foundation for further analysis. Next, we move on to data pre-processing. This crucial step ensures data quality and integrity. We correct baselines, detect peaks, filter noise, and align retention times across samples. By doing so, we remove any variations or artifacts that could affect the accuracy of our analysis. To ensure the reliability of our analysis, we assess the quality of our experimental data. We examine factors such as reproducibility, instrument performance, and overall data quality. This assessment helps us validate the robustness of our results. Now let's dive into data analysis. Our first task is the identification and quantification of lipid species. We compare our acquired mass spectra with known lipid species in reference databases. This allows us to identify lipids and measure their abundance. We explore the composition of lipids within our samples. We analyze the distribution of lipids across subclasses, such as phospholipids, glycerolipids, and sphingolipids. This helps us understand the diversity and complexity of lipid profiles and their metabolic pathways. We also investigate the dynamic distribution of lipids. By studying changes in the relative abundance of specific lipids across different conditions or time points, we gain insights into lipid metabolism, cellular responses, and disease-related alterations. One important aspect of lipidomics analysis is the identification of differentially regulated lipids. We compare the abundance of lipids between sample groups to identify significant changes. Statistical techniques such as t-tests or ANOVA help us determine the significance of these abundance changes. Furthermore, we explore alterations in chain length and the degree of saturation within lipid acyl chains. These parameters play a crucial role in lipid function. Statistical methods allow us to assess the significance of these changes between sample groups. Finally, to gain a deeper understanding of the biological implications of our findings, we employ pathway analysis. This involves mapping the identified lipids onto relevant metabolic pathways using tools such as KEGI or MetaboAnalyst. Pathway analysis helps us unravel the complex biological processes underlying lipid-related diseases. The comprehensive understanding of lipid profiles and their functional implications opens up a world of possibilities for advancing research and practical applications. One prominent application of lipidomics is in the field of biomarker discovery. Lipidomic analysis enables the identification of specific lipid signatures that can serve as diagnostic or prognostic markers for various diseases. By analyzing lipid profiles in different patient populations or experimental models, researchers can uncover lipid biomarkers that aid in early detection, disease monitoring, and personalized treatment strategies. Lipidomics also plays a crucial role in understanding the pathogenesis of complex diseases. By examining alterations in lipid profiles associated with different disease states, researchers can gain insights into the underlying mechanisms and pathways involved. This knowledge can contribute to the development of targeted therapeutic interventions and the discovery of novel drug targets. Moreover, lipidomics has found applications in pharmaceutical research and drug development. By analyzing the effects of drug compounds on lipid profiles, researchers can assess drug efficacy, identify potential side effects, and optimize therapeutic interventions. Lipidomics serves as a valuable tool for understanding drug-lipid interactions and guiding the development of lipid-based drug delivery systems. Lipidomics has proven valuable in the field of nutrition research. By analyzing lipid profiles in response to dietary interventions, researchers can elucidate the impact of specific nutrients and dietary patterns on lipid metabolism. This knowledge helps in designing personalized nutrition plans and optimizing dietary recommendations for improved health outcomes. Another fascinating application of lipidomics is in the field of environmental toxicology. Lipidomic analysis allows for the assessment of lipid profiles in response to environmental contaminants or pollutants. This helps in understanding the impact of these substances on lipid metabolism and cellular health. Such insights contribute to environmental risk assessment and the development of strategies for pollution mitigation. Creative Proteomics is your premier partner for lipidomics analysis services, offering both untargeted and targeted approaches to meet your research needs. Our advanced analytical techniques, state-of-the-art instrumentation, and experienced team of scientists allow us to deliver comprehensive and precise lipidomic analysis. With our untargeted lipidomics services, we can provide a global view of the lipidome, capturing a wide range of lipid species and uncovering potential biomarkers or metabolic pathways of interest. Our targeted lipidomic services enable a focused analysis of specific lipid classes, subclasses, or individual lipid species, providing detailed insights into specific metabolic pathways or targeted investigations. We can analyze various lipid species, including but not limited to this table. Our team is dedicated to providing accurate, reliable, and actionable results, assisting you in your lipidomics research projects. We work closely with our clients to design customized experimental protocols, optimize workflows, and ensure data integrity throughout the analysis process. Thank you all for joining us today and learning about the exciting field of lipidomics. If you have any further questions or would like to know more about Creative Proteomics and our lipidomics services, please visit our website or contact us. We look forward to hearing from you.